Probably not going to get through all of photosynthesis. I will warn you that photosynthesis is a bit of a heavy topic, particularly for anybody who's not really seen it in any detail before. The other thing to know about photosynthesis is I'm not going to cover everything about it. So there are things in the notes that you're not going to see in these PowerPoints and so on. But if you have any questions about it, let me know. It's one of those things that the more you learn about it, the more interesting it becomes. If it's just your first time through, it may not seem so exciting, but it is something that does get really cool if you learn a lot more about it, particularly what's going on in bacteria too. We're going to be mostly talking about what is going on in plants. So this is, of course, not photosynthesis. This is respiration. This is what we were talking about last day. So respiration, we start off with some sort of organic molecule. And specifically, we were talking about glucose last time, and it gets oxidized. So that requires oxygen. And so during this process, it gets broken down into carbon dioxide and water. And if we produce ATP as kind of a bonus is what we're trying to do. So that's our respiration. That's going to happen in your mitochondria or the mitochondria of any other species that has one out there. Photosynthesis is kind of the reverse. So you can see we're starting off with carbon dioxide and water over here on the left. And we're going to create organic molecules, and we're going to release oxygen. And this, of course, doesn't take ATP, it takes light energy instead. That's the photo part of photosynthesis. So some people will talk about how they are the same reaction, but in reverse, which isn't quite true because, of course, photosynthesis isn't consuming ATP. And when you start to look at the details, it's a little bit more complex than that. But you're going to see a lot of parallels. We have oxidations going on, we have electron carriers, we have electron transport chains, and all those things are similar between photosynthesis and respiration. So one thing we're going to do when we review for the midterm is kind of come back and revisit some concepts around photosynthesis and respiration as well. So we'll get there. Today we're talking about photosynthesis. And first thing to know about photosynthesis is that photosynthesis is done by organisms that we call autotrophs. So auto means self, and troph means feeding. So these are things that are self-feeding, means they make their own food, and they're going to do that by absorbing energy from the sun or other light sources and produce their own organic molecules. So something that I've done before, I've asked on midterms, who does photosynthesis? And of course, the obvious one is here, plants. Don't forget all the other possible autotrophs out there. We have unicellular protists, so this is like euglena there. We've got multicellular algae, and there's different types of bacteria. You can see there's the cyanobacteria, they're green, but we also have these purple ones here, these purple sulfur bacteria that actually produce elemental sulfur as part of their photosynthesis process. So don't forget, it's not just plants. We have talked about two types of chloroplasts in the lab in this course. There's the Elodea on the left, that's the aquarium plant. We also saw the Protist euglena, which is shown there on the right. And I know in the lab videos and images, sometimes it's hard to see the chloroplasts, and I found this diagram here, which is really good. You can actually see the chloroplasts in a little bit of detail there and have an idea of what they actually look like in there. This week in the lab, we're going to talk about a third type of chloroplast. We're going to talk about spinach chloroplast. So that will be on Thursday's lab. Here's some electron micrographs. You can see kind of like the mitochondria, chloroplasts have multiple layers of membranes in them. So that's shown there in that SEM. There's another one, you can see maybe the folds of the membranes a little bit more clearly. And depending on the species, chloroplasts can look actually very, very different from one another. They're generally a lot bigger than mitochondria, 2 to 5 times the size. And here's another diagram. This one is from the textbook. And just a reminder that since they're making carbohydrates, they can also store some carbohydrates. So in that case, you can see the starch is stored there. And there's a chloroplast from a red algae. So the red algae must be red, but the chloroplast itself is green, I'm guessing. I'm not entirely sure about that. Remember, this is a an electron micrograph, and micrographs from electron microscopes are not actually in color. We have to add the color later. So I don't know a lot about red algae, but apparently they're very interesting organisms. So what if you are photosynthetic and you're a bacterium and you don't have organelles? Well, there's lots of bacteria like that, like the cyanobacteria, and they do have extra membranes. So we don't necessarily call them organelles, but basically it's the plasma membrane is folded in on itself to add all those extra layers, and that's where they're going to have their photosynthesis and their chlorophyll. So let's talk about these chloroplasts. Usually I draw one on the board. You don't need to make yours so fancy. What you're looking for is basically two membranes and then these little pancake things in the middle. So usually mine looks something like that, a little bit messier. So kind of like what I did with the mitochondria, I want to just label a few things here and kind of talk about what's going on in those locations. So like the mitochondria, we have two membranes. One is called the outer membrane, and the next one is called the inner membrane. And I don't actually have much to say about those membranes. They are mostly structural, holding the whole thing together. We do have another membrane here, and these ones are colored green for a reason, and these are called the thylakoid membranes. Thylakoid membranes. So they are green, like I said, for a reason. And so this is, of course, the location of the green stuff. And the green stuff is, of course, chlorophyll. So location of chlorophyll. And since we have chlorophyll there, this is the location of the light reactions. So that's an important part of photosynthesis. So there's a couple other places to label. One is the inner section of these thylakoid membranes, and this is called the thylakoid space. So what goes on in the thylakoid space? This is the location of the proton motive force. So remember, we talked about the proton motive force. This is what drives ATP synthesis. So we are going to have some ATP synthesis here. That's actually an important part of photosynthesis, is making a little bit of ATP. It's not an end product, it's kind of somewhere in the middle. It's an end product of the light reactions. So there's one more place to label, which is this part between the inner membrane and the thylakoids, and this is called the stroma. So what goes on in the stroma? So this is the location of your DNA. So remember that chloroplasts have they have DNA and ribosomes, and that's where that is found. This is also the location of the Calvin cycle. So this is a biochemical reaction that is still named after the person. So the Calvin cycle is also known as carbon fixation, and we'll talk about that as well. And one more thing to say, since this is where the sugar is made, is that this is the location. I'm running out of space to write. It's not wanting me to write down there. Let me just see if I can squeeze it in. Location of starch granules. There we go. I can just fit it in. Because that's the final one of the final products of photosynthesis is, of course, making those carbohydrates. And they sometimes get stored there as starch. So hopefully you got all that. You can always go back and review the video if you need to. Inner membrane, outer membrane, thylakoid membranes, thylakoid space, and the stroma. There's an R right there. Not stoma, but stroma with an R. So there's one of these fancy pictures of chloroplast. You can see the hybrid electron micrograph on the left and the cartoon on the right. You can see all the stroma is labeled here. We've got the thylakoids. A stack of these thylakoids is called a granum. You don't need to know that word, but if you're interested in what a stack of them is called, there you go. It's not really so important to understanding photosynthesis to know that what a stack of those is called. So one other term that's important to know is this term here, a plastid. So what is a plastid? Because sometimes people will say plastid when they mean chloroplast. So plastid is basically a pigment-containing organelle in a plant, usually. Pigment-containing organelle. So the reality is when people say plastid, 99% of the time they mean chloroplast. But every once in a while, they are talking about a different kind of organelle. So take a look. There's these starch granules. They're in potatoes. You may have noticed that some potatoes have different colors. Some are kind of yellowish, and some are kind of purplish. They have pigment in them, and that pigment is an organelle, and it's a type of plastid. It's not a chloroplast. Here's another example on the right. This one's usually more obvious. I ask people, what part of a plant is colored? And people say the green part, and I say, what about other plants? And some people say flowers, and of course that's what we're talking about here, is the pigment found in different types of flowers can be found in plastids. So if you ever see that word plastid, this is what it means. It means, usually it means a chloroplast, but really the best definition is that it's a organelle that has some sort of pigment found in a plant. So let's talk about the reaction of photosynthesis. So we already talked about this, that we're going to take some carbon dioxide, we're going to take some water, we are going to require some energy in the form of light, then we're going to make some sort of carbohydrate, and we're going to release oxygen. Notice it also produces a little bit of water, but it takes more water than it produces, so often we don't include that. Again, I'm not so concerned about the balancing of these equations, more or less thinking about what is going in and what is coming out in the net form of the whole thing. So I want to talk a little bit about these reactants before I talk about photosynthesis. And the first one to talk about, of course, is light. And probably most of us know what light is, unless you can't see, of course. But light is what makes things colorful. And it's a type of radiation. And by radiation, it means it's something that has energy in it. So if you know a little bit about the electromagnetic spectrum, there's all sorts of different types of radiation. So you've got some high energy stuff over here, gamma rays and X-rays, and these things can give you cancer. UV rays can as well. They can create a lot of heat, and they can actually damage DNA. And then we have the visible part of the spectrum, and this is where we have all the colors that we love and know, ranging from violet and blue all the way to red. And so this means that violet and blue has actually higher energy than red and orange down on that end of the spectrum. And then, of course, we've got infrared and microwaves and radio waves, and those things have their uses. If you like your cell phones and things like that, that's in the radio wave to microwave region of the spectrum, by the way. So there's energy there, and that is going to be absorbed by pigments in plant cells and other autotrophs. So you're probably familiar with this one the most, chlorophyll. Chlorophyll, of course, is green, and that's what it looks like. But there are other pigments out there. You probably notice them in the fall time when chlorophyll starts breaking down, and the other pigments are a little bit more stable, and so we see all sorts of interesting colors. A little bit more yellow and orange around here in this part of the world than other places, but sometimes we see some interesting reds in maple trees and whatnot, and these are all different pigments. So take a look at these pigments, and you can see one thing that I noticed right away when I look at the structures of them is they are loaded with double bonds. And when you have these systems of double bonds, these are actually called conjugated double bonds, where you can see if you take a look at this flavanol, I've got a double, a single, a double, a single, a double, and so on. And so when you have double, single, double, that's a chemistry term for, it's called a conjugated double bond. And anytime you see a lot of those, often these are molecules that absorb really well at the visible part of the spectrum. And so you can see these ones here, they look different colors. and you might notice them, like I said, in the plants out in nature. So we can do better. We can figure out what color these things absorb by using a spectrophotometer. So you may remember way back in lab 3, we used a spectrophotometer and we were looking at the beta cyanin, the purple color found in the beets. And we were measuring, I'm trying to remember, were we measuring 525 nanometers maybe? So you can do this thing where you can do something called an absorbance spectrum. So you can see that word is over here. And this is where you take the spec and you take a reading at every wavelength. So if you do this for the chlorophylls, starting at 400 to 700, that's most of the visible range. So if you take a look, I'm looking at chlorophyll A right now. You can see it absorbs really well here, kind of in the purplish bluish part of the spectrum, and then absorbs again really well here, somewhere around 670, kind of an orangish reddish part of the spectrum. So chlorophyll, and you can see this is true, also true for chlorophyll B, absorbs really well in the blue and red regions, and not really well in the green part of the spectrum. So what does that mean? It means that if it's taking and absorbing those colors, then green is what's left over. And that's why chlorophyll looks green. If you look at the carotenoids, it's absorbing really well just in the blue and a little bit in the green sort of area of the spectrum, but not at all in the red and yellow part of the spectrum. So the carotenoids, they look red and orange and yellow. And that's how absorbance works. So we are going to produce an absorbance spectrum, by the way. That's one of the exercises we are going to do for lab 7 of the spinach chloroplast. The second part of experiment 7, by the way, we are going to do something called an action spectrum. So an action spectrum means we are actually going to measure the chemistry and the action of photosynthesis at 3 different wavelengths. So if you take a look at this, this is showing for a typical plant. We have a lot more photosynthesis action in the blue part, just a little bit in the green, and again, a bit more in the red part of the spectrum. So what we're going to do is do these kind of spectrums to confirm that we're seeing more absorbance and more action in the same colors and parts of the spectrum. So more on that on Thursday, by the way. So here is chlorophyll. Let's talk about how chlorophyll works. This is a little model of chlorophyll. You can see it looks a lot like, almost like a tennis racket. And I kind of think of that as a good analogy because what it is doing is catching photons of light. So as I mentioned before, chlorophyll has all these conjugated double bonds, and that's a really good way to capture light energy. And there's actually 2 chlorophylls in plants called A and B, chlorophyll A, chlorophyll B. They have slightly different spectrums of absorbance, which is good because they're trying to capture more light and more energy. So if one of them captures light in one area, chlorophyll B can capture light in another area a little bit better. So chlorophyll is going to capture light energy. And we're going to talk about a little bit how that works in a few minutes. I'm just going to show you this. Chlorophyll is a very hydrophobic molecule. And probably remember, hydrophobic often has something to do with membranes. In fact, it is. Chlorophyll is bound by these proteins. These proteins are sometimes called photosystems. And these proteins are embedded in the thylakoid membranes. And so those thylakoid membranes are green, as I pointed out, and that is where all the light part of photosynthesis, where the light is going to be captured there. So I'll come back and talk about how that works in a minute. So we have light, we have carbon dioxide. Carbon dioxide, of course, is a product of cellular respiration, also a product of human activities such as burning fossil fuels. And so there's plenty of it in the air in most places, and it's going to go into the leaf and be part of photosynthesis. And the last part is water. So water, of course, comes from the ground. Plants usually absorb water through their roots, and it's an important part of photosynthesis. Okay, so let's talk about the reactions. We have 2 parts. We have the photo part, and we have the synthesis part. So let's take a look at that photo part first. So the photo part, of course, photo means light. It's going to involve light. It also involves water. So those are the two main ingredients for photosynthesis. We also have this NADP+, I'll talk about in a moment, and ADP and PI. So we'll get to that in a moment. But kind of the main thing going in here at this stage is water. So water is actually going to get split. And I'll explain what I mean by that in a moment. Well, maybe I'll write you the formula for that. Water is going to get split, and it's going to get split into hydrogens plus oxygen plus electrons. So we have seen that reaction before. Remember, we were using oxygen in respiration. Now we're kind of doing the opposite. So notice the splitting part, what the products are. We've got electrons and we've got oxygen. So that's really what's going on here, is we're taking electrons from water. Those electrons are taken from the bonds that are binding the oxygen to the hydrogen. And the result is we get oxygen. So the oxygen at this point, I hate to use the word waste product because it's good, we like oxygen. So maybe byproduct is a little bit better, but it's not used again for photosynthesis. Now that same plant may take that oxygen and use it for its own respiration. Plants do respiration as well. Don't think it's an animal plant thing. Plants also do have mitochondria, and so they will use the oxygen. But for the most part, it's going along, and us as animals, we're very happy because we have that oxygen. So notice the other things that are going on. The question is, what happens to those electrons? I was telling you last week, electrons, they don't usually like to be just free living out in the world, not attached to something. They're going to attach to new bonds, and notice we're creating some new bonds. So we're taking NADP+, so that's very much like NAD+, except for it has an extra phosphate. And it's going to add a bond here. So that H right here is actually a hydrogen, and now it's attached through electrons in that bond. The other thing we're doing is we're going to make a little bit of ATP in this process. So ATP, to make it, we need ADP and inorganic phosphate. So all that energy from the light takes electrons from water, and it puts them in new bonds, high energy bonds in ATP and NADPH. And that's it for the light part of photosynthesis. We'll look at it in just a little bit more detail in a moment here, the light reactions. Then we have the synthesis part of photosynthesis, and it's called the Calvin cycle. And so what happens with the Calvin cycle? These high energy compounds here, they're not very stable and they're not very long lived. So if you want to store energy for a while, the best way to do it is to store it in organic compounds. So how do we make organic compounds? Well, we need carbon. And so plants get carbon from carbon dioxide. So this is called carbon fixation, meaning we take carbon from carbon dioxide and we make new bonds, and those new bonds end up in organic molecules such as sugars. And so the ATP and the NADPH are recycled, meaning the ATP is broken down into ADP and PI, and NADPH is broken down into NADP+. So I'm going to put a big star here. Learn this slide for sure, 100%. If you need to know something about photosynthesis and you can't remember anything else, learn these reactions that you see here on this slide. Everything else is just details, but if you need to start somewhere, this is where you got to start. It's got lots of good information here. It shows you where the light reactions are, shows you where the Calvin cycle is, shows you the products and reactants of both light reactions and the Calvin cycle. So we're going to take these apart a little bit and discuss them in a bit more detail. Starting with the light reactions, I don't know if we will get to the Calvin cycle today, but I do want to talk about the light reactions today for sure. And there's a lot of details there, and I'm bringing you through these details. I'm not going to ask you on a test to regurgitate all of the light reactions of photosynthesis. That would just be nasty. But I do feel it's important to go through the details to kind of get an understanding of some certain things here that we're going to talk about. So let's take a look at these light reactions. So there's a note that says the NADP+ and ADP are recycled. So they're not part of the overall reaction of photosynthesis because they're kind of recycled. But they are part of both the light reactions and the Calvin cycle. They're intermediates that are short-lived. and sugar is made from light. Okay, so let's talk about these light-dependent reactions. That's actually a better way to describe these reactions. Sometimes people call them the light reactions, but light-dependent reactions is probably a better way to describe it. So what you're seeing here is a chlorophyll molecule. And some of you probably took Chemistry 101, and maybe you remember sticking different things into Bunsen burners, and they light up and make some nice colors. It's kind of the same principle. What you have here is light is getting absorbed by that molecule, and the electrons, they get excited, right? They go into higher orbitals. And sometimes the electrons get ripped off, sometimes they go back to chlorophyll, and when they go back, they will release more light, okay? So this is kind of what's happening with chlorophyll. And like I said, you can think of chlorophyll as a big tennis racket, but picture it as like a whole bunch of tennis rackets. And the ball that's coming over the net is probably going to hit one of them if you have a whole bunch of them. So we call this whole thing here a photosystem or a reaction center. And so that's what I'm trying to show you here, is that we have a whole bunch of chlorophyll molecules, a whole bunch of them. So the light comes along, and there's a good chance that it's going to hit at least one of them. Now, it may not hit the right one, and that's okay. And then the photons, the light, just sort of bounces and goes from one to another, and eventually it hits something called a reaction center. And at that point, those electrons are going to enter the photosynthesis system. I'll talk about that in a minute. So if you see this term here, photosystem, it really just means we've got the pigments, so the chlorophylls and the carotenoids, and also a bunch of proteins. So that's just a term for what you're seeing here, is this big complex. It's a whole bunch of chlorophyll held in place by proteins, basically. So here's just another picture. I gave you the tennis racket sort of analogy. This one's sort of making it look like a big funnel that's going to capture all this light and eventually send it down to a reaction center way down here. So talk about this reaction centers in a moment. So let's talk about these light reactions. We're going to break them down into two parts, and we're going to call it photosystem 2 and photosystem 1. So the confusing thing here is that notice photosystem 2 is first, and then we have photosystem 1. So why is that? Well, it turns out that photosystem 1 was discovered first, and then we discovered photosystem 2, and then we realized that they went in this order. So unfortunately, the name has stuck, although you will see sometimes people call them P680 or P700, and that's kind of just describing the best wavelength of light that each of them absorbs. So there's a whole bunch of steps here, and I'm going to break this down over the next few slides. But very briefly, I'm going to tell you what's happening here, and then we'll go over it again. We've got light hits the chlorophyll, so hopefully that part is obvious. But then what happens? Like I said, we've got electrons, and electrons are eventually going to get ripped off some chlorophyll, and I'll come back to that water in a second. And they're going to go through this system, and you know what happens with electrons that go through systems? Sometimes we have electron transport chains, and electron transport chains make ATP. And sometimes those electrons end up on electron carriers, and that electron carrier over there is NADPH. And those things are important for the Calvin cycle. So that's kind of the light reactions in a nutshell. The last part is just the water part. So remember I said the electrons are getting ripped off chlorophyll. Well, that would destroy the chlorophyll if we kept doing that. So we actually replenish the electrons from water, which also makes oxygen as once water gets stripped in half. So let's take a look at these light reactions kind of step by step. And I'm going to write a few notes along here. I think I have some notes in the PDF files that I provide you guys, which will be relatively similar. But I kind of like to write this out as we go along just to kind of understand what we're doing here. So if you take a look, there's number 1. So number 1 is basically light hitting chlorophyll. So light hits chlorophyll. Okay, that's great. And then you can see chlorophyll eventually loses those electrons. And so maybe I will say that as number 2. Or maybe number 1. Let me think of how to write this. Light hits chlorophyll, and maybe we'll say, and eventually and that will be number 2. Eventually electrons, so that means electron, the E with the negative beside it, are donated to an electron transport chain. Are donated to an electron transport chain. So that's it. Light hits this thing, it gets warm, and the electrons get ripped off and go to an electron transport chain. Great. Okay. 8 steps. So there's 1 and 2. Light hits this, it gets warmed up, is what goes on. Number 3 is where we split water. So we got to say that water splits. Okay, so let's take a look at the reaction of water splitting. We have H2O, and this is a balanced equation. That's why I have the 1/2 O2 there. And we have some protons, and we have some electrons. And these all go to different places. So the O2 is our byproduct. The hydrogens, these are going to get pumped by the electron transport chain. And those 2 electrons, maybe we'll just go a little bit lower. So these replenish lost electrons from chlorophyll. So that's number 3. So at this point, we've warmed things up with a little bit of light. Those electrons have gotten excited. They moved on to an electron transport chain, and we just replaced them using a little bit of water, because water is a handy and common substance, so it's easy for plants to use. So number 4. Number 4, we have an electron transport chain. What do we do with electron transport chains? So you probably know electron transport chains are moving electrons in bonds from protein complex to protein complex. But the main thing they do, electron transport chains, they pump hydrogen ions and they go into the thylakoids. And so this produces a proton mode of force. And then what else happens? We have a proton mode of force. What is that good for? A proton mode of force is good for number 5. And number 5 is an ATP synthase makes ATP. You can say it makes ATP from ADP and PI if you want. That much is, in my mind, relatively obvious. So this isn't too bad. We're actually making a little bit of ATP, and the cell in some cases can make use of this ATP. But like I said, ATP is a short-lived molecule, and if you want to store energy long-term, or if you want to just build things, you want to build carbohydrates or whatever, you're going to have to eventually transfer those electrons to new bonds in organic compounds. So let's talk about 6, 7, and 8 then. So 6, actually, I'm just going to put it all in one slide here, and I'll have to write over here in the corner. So this is where we start to get the advantage of being a plant. It has 2 photosystems, so it's just trying to be a little extra efficient. Like I said, these photosystems 2 and 1, they absorb at different wavelengths. So photosystem 2, you've taken those electrons, we've exhausted them, taken away some of the energy, we've made some ATP. And what we're going to do is keep using those electrons. We're going to use some more chlorophyll, and we're going to use a little bit more light. So that's what number 6 is. So number 6 is light hits more chlorophyll. So light hits more chlorophyll. Okay, and so what happens after that is number 7. So I'll do number 7 up here in the corner. And now we get, we have another electron transport chain. So I'm just going to put the arrow here. And electrons are donated to an electron transport chain. And I'm not going to have a big explanation about this, but basically those electrons are replenished from the first photosystem, photosystem 2. Kind of the main point here at the end is what happens to those electrons in the end. So in the end, those electrons, they reduce NADP+, and they reduce it to NADPH. And that's kind of it. So I know there's a lot of steps here, but I just kind of wanted to break it down a little bit and kind of, show you what is going on here, because there's a lot of familiar concepts. We have electron transport chains, we have electron carriers, we have redox reactions and those kind of things that are very similar to what we've seen in respiration. So this is just a little diagram from the textbook to try to show you kind of an idea of those electrons getting more or less potential energy. You can see it's the light is giving them potential energy. They go up, and as they go down, their energy is converted to bonds in ATP and bonds in NADPH. And so here's another little diagram here. And you can see this is just kind of showing it all in the context of the thylakoid. And the protons, oopsies, the protons are all right here. There's my proton motive force. And they're going to go out here through ATP, and they're going to make ATP in stroma, and the NADPH in the stroma, and this is all going to go Kelvin cycle. So a couple more things to say here. You may have noticed that on my slides I called it linear electron flow, and that means the electrons, they have a beginning and an end. This is kind of a footnote, I'll never ask you about this, but this is in the textbook, talks about cyclic electron flow as well. And cyclic electron flow is a way for a plant to kind of just recycle its electrons, not put them on NADPH, to kind of make extra ATP. Extra ATP is important because it's a cell, it uses tons of it. Okay, so there's our ATP synthase. It's the exact same ATP synthase that we saw in the mitochondria, and it's making ATP using a proton motive force. The only difference is the labels. We have a thylakoid here in a stroma because we are in a different place in the cell. So back to this slide here. Oops, sorry about that. That is a hint for next day. I'm going to stop at this slide because I have Ruth on the line here. She wants to do the class evaluation. So I'm just